Hi, everyone. Welcome to the Small Cap Discoveries conference call. Today on our call, we have Dr. Jennifer Bath, the CEO of Immunoprecise Antibodies. Immunoprecise Antibodies trades on the TSX Venture Exchange under the symbol IPA and on the OTC under IPA-TF. The company is trading at $2.60, a new all-time high, with roughly 88 million shares outstanding, including all the recently exercised warrants, or about a $230 million market cap. I'd now like to hand it over to Paul Andriola.
Thanks, Trevor. I'm excited. I know a few others on the call here are pretty excited. We probably the most anticipated presentation we've had in a long time. I'm happy to have Dr. Jennifer Bath with Immunoprecise. Before I go any further, I have to disclose that not only am I a director, but I'm a very significant shareholder in Immunoprecise, so I wanted to make sure everyone was fully aware of that. But Jennifer, thank you for joining us. Exciting times at Immunoprecise, and obviously we'd love to get updated and get a good understanding of what we can expect going forward.
Absolutely. All right. Well, thanks everyone for joining me today, and it's certainly my pleasure to provide you with an update on Immunoprecise and its activities, and also our strategies. As most of you on the call today have probably heard our history, and some have even probably lived through this with us, I thought I would focus a little bit more on our long-term strategy behind some of the decision-making that we've made and how it ties to where we are today, and also where it is that we are focused on going. So referencing our disclaimer slide, please note that throughout today's call, I will be making some forward-looking statements, and also that these statements are based on our beliefs and also our expectations, and are subject to revisions and also to some uncertainty. Okay, so most of you know already, we in 2016 set ourselves on quite a very different trajectory, making a very purposeful pivot in our strategy, specifically toward the $30 billion therapeutic antibody market. And after identifying significant deficits in the market, we set out to disrupt what is known as the traditional multi-vendor drug discovery process by addressing the two greatest challenges that really face that market. The first was to unveil cutting-edge technologies to end the expensive and time-consuming repetition in studies. Quite literally, what we were hearing repeatedly and what was being shown in various market analyses was that pharma were tired of the repeated and failed attempts to find good clinical candidates when they were using CROs with antiquated technologies. The second objective, which we've also now accomplished, was to become the only CRO to bring this full suite of services all in-house, greatly reducing the risk by eliminating the traditional multi-vendor model, but also enabling the transparency of global work from one centralized location. So as you can see from our historical timeline here, we've been dedicated to these goals for over 2 years now. We've accomplished these objectives primarily through strategic investments in R&D for new technologies, as well as through strategic acquisitions. And we remain focused and dedicated to always pushing our technology through innovation to really set an industry-leading standard and to maintain and keep that industry advantage. All right, so I put the history slide before the management slide today because I wanted to give you some context as to how my vision impacted our strategy and also my decision-making around building a new management team. During the strategic growth period, we also had a tremendous lift in building the company and the company infrastructure, which would enable all three of our subsidiaries with their technologies to tie together seamlessly, also catalyze further growth, and then also represent one unified global company. And so, one of the things that was key to this effort for us was obviously the selection of a formidable executive team. I needed well-respected global leaders with track records in building successful CROs, and in tune with the market and with the newest technologies. And as I've already indicated to you, those didn't always go hand in hand in our market. And so these are really unique individuals that I really set out to bring in. And then also individuals with experience in moving R&D candidates through clinical and commercial successes. So, you know, a lot of different criteria, a lot of different hats, and going out and really seeking an executive team that could really help push the boundaries and build something that would really revolutionize this space. So on this slide, what you're really looking at as essentially what we set up here to bring something completely unique forward to this industry, and exactly you know, what we built to change the standard in antibody discovery and development today. We have fully human samples at the ready, which we can use to exploit the full human antibody repertoire. And that's without the need or the wait for patients to recover from a disease. And so obviously, you know, with respect to COVID, I think that's something a lot of people can appreciate the value of. In addition to that, we have proprietary and optimized protocols across all of our discovery platforms for use with leading transgenic animals. In addition to that, IPA runs multiple parallel and automated platforms, and many of these, and I think this is something that oftentimes people don't realize, many of these have the capability of being monitored remotely around the clock. So it is really a unique aspect of what we are able to do when we need to do, for instance, expedited protocols, or just make sure that we have an extra set of eyes on programs that are exceptionally large in scale. Our B-cell platform, which I know is a favorite within our investors, and it's also definitely a favorite with our clients, it has the ability to screen over 10 million blood cells per run, which has the potential to result in thousands of primary candidates coming out of that platform. And that's compared to, you know, it's thousands compared to hundreds at the most coming off of traditional technologies. I think, you know, actually with respect to this, I'm looking at this slide, one of the things that I think is perhaps most interesting here is that we were able to pressure test, you know, these technologies as we brought them forward. We put them all together under one roof and integrated them, and then we were able to pressure test them and demonstrate the capacity and the capability working with some of the world's largest pharma. And we offered those technologies up to them to solve any target class, regardless of the disease, regardless of the protein family and how challenging that target might be. And as you can guess, you know, when pharma companies came in the door, they didn't bring us their easy programs. They didn't bring us the things that they thought, you know, oh, okay, well, you know, this one's easy, so I'll let you do it. We'll take the challenging one. They brought us the programs that they struggled with. So after, as many of you know, I'm sure, you know, waiting and looking to see how this would come together after substantive audits and negotiations, we have now successfully used our technologies to contribute to building the pipelines of 65% of the world's top 20 pharma and over 450 clients worldwide, with very high success rates in many of our products that were moved on actually toward further clinical development. So here's a glimpse into our pipeline, which I'm sure you know is housed at Talem Therapeutics. Our pipeline that we developed this past year, I think, is an exceptional example of the benefits that we gain by using the same technologies we leverage for our pharma partners to build their pipelines. But also, and I think this is interesting to point out, because I had a great call with an investor, you know, a few months ago, who said, gosh, you guys are kind of, you know, you're a little small company. What makes you think you can compete with these big, you know, Mercks or you know, Janssen's or whatever of the world that are out there also working in COVID? And it was kind of interesting because, you know, obviously I pointed out, yes, okay, many of these groups are using us. They're using, you know, our technologies to build the same pipelines, but they're also using our same scientific, you know, teams, right? The same scientific teams that we have at our subsidiaries. They're the ones that design those programs, build those programs, and lay out how those programs will be accomplished on those same technologies. And then we also have the added benefit of our talented Talem scientific committee, and then also our experienced executive team. So also with respect to this pipeline, which I think is important, because if you go and you investigate other, kind of smaller and mid-sized companies on the TSX and on NASDAQ, you'll notice that oftentimes, you know, they have a directive to be focused on a particular disease. We are disease agnostic at IPA, and what we look at when we go into a program is, you know, we look specifically in terms of when we, you know, when we investigate a target, we look specifically in terms of the market. We look at the need, so, you know, the number of patients, and the impact on those patients. We look at what's called the druggability of a target, and then also the availability within the patent space, which is obviously really important before you begin a program. We address any protein and any target class. We also do that with speed, and I think also very importantly, and probably not lost on investors, is that we can do this at cost. IPA does retain 100% of ownership of every asset, so we don't lose time or also focus, I think is important in negotiations with partners. And, you know, obviously within this pipeline is also where we house our COVID vaccines and also our therapeutics for COVID. So going back to January, about mid-January of this year, that's when we launched our anti-SARS program, what we believe is the most comprehensive and rigorous anti-COVID program that exists today globally. We expressed concern early on about the political and financial incentives, emphasizing the speed in the development of vaccines and therapeutics. I know that we, you know, we discussed that publicly in our releases starting around February. And not only the concerns around the speed in the development and the pressure from political realms and financial incentives, but also the compression of the timelines for unvented platforms and enabling the advancement of trials prior to proper regulatory review. And we had a nice little roundtable kind of fireside chat a couple weeks ago. Maybe you were able to attend that, you know, where I addressed some of those concerns and some of the challenges we have now today because of trials moving forward without proper regulatory review or proper preclinical data vetting. So we brought forward our Polytope approach sometime around mid-February. We were fully aware that we were uniquely positioned to launch a program with the scale and also the scientific rigor that was required to produce the diversity of lead candidates that were needed to create a cocktail therapy. And this is widely believed to be the most efficacious approach to addressing emerging infectious diseases, because preventing virus escape through mutations, especially with an RNA virus, is a very well-documented phenomenon. And so the breadth and the scope of our program going into this needed to be exceptionally broad to ensure that at the end of that cycle of those programs, we had enough quality and diverse antibodies to really put forward meaningful cocktails to address you know, this particular emerging RNA virus in a cocktail format. So our final output, you probably read earlier this spring, we had approximately 75 potent neutralizing antibodies, which means that they're able to prevent the virus from entering human cells. This was not only the most globally comprehensive panel that we're aware of, but it also resulted in a very diverse set of panels of therapeutics to enable the types of diverse cocktails that we're preparing in-house. And also, you know, supporting the development of safe, efficacious, and sustainable therapies to protect and treat against all current and future strains and variants of SARS. All right, so just a little bit about our Polytope monophonal antibody therapy. So we have, you know, building this extensive platform that we use for this approach. It requires not just, you know, high capacities and powerful technologies. It does also require the experience from our teams in developing clinical products to emerging viruses. They're very unique in multiple aspects of the way that you go about addressing these types of programs, and also very in-depth analyses of every candidate along the way, and a comprehensive characterization of those candidates to really understand every detail of them, every detail of how the antibody binds, how it interacts with the target, how does it interact with other antibodies, how well will it manufacture, how stable is it, you know, what is its function? Does it have the ability to, prevent infection? But more than that, you know, we should understand in this, type of a situation where you're preparing a product to go into human, it's more than does it protect? It's all the answers around how does it protect? What exactly is it doing? You know, those are the things that really enable a safe and efficacious development of a therapeutic. And those are the types of attributes that we screen for when we specifically have that objective. So on this slide, I want to cover just some of the advantages of using the Polytope, because I think, obviously it has huge potential with respect to addressing SARS-CoV-2, but the potential definitely doesn't stop there. So, we've obviously used these platforms in addressing COVID, and we do routinely use these platforms with great successes against many other diseases. We've really revolutionized the process for our pharma partners. And I think here's kind of the take-home message with that. I mean, we have these assets that we've now developed that are very well poised to address not only the SARS pandemic and future variants and strains of the SARS pandemic, but we also have a multitude of assets that are exceptionally promising in the areas of oncology, of inflammation, and also rare diseases. And in addition to that, we have these vast amounts of data that we've collected on SARS-CoV-2 that keeps us very well positioned to continue to treat future strains and variants of the virus. And that's something that we're very actively engaged in and working with, multiple government groups to talk about those strategies and how do we use them, to really make sure that, now with that information, we can address this rapidly, but without sacrificing quality, right? That's the most important part. With all the data there, this now becomes a plug and play system to ensure that, going forward, you have that same quality and that same tendency towards safety and efficacy, but able to address now new strains and variants in an exceptionally rapid way. And all of that goes back to, how we start off with this in the beginning, with a broad and scientifically rigorous and purposeful program. And then, this lastly, I do want to address the fact that this, Polytope technology is very well positioned also to just change the way that we address emerging infectious diseases in general, and also the way that our partners do, with unparalleled scientific rigor and enhanced timelines, and also, most importantly, being able to do that, with those timelines, with that scientific rigor, while still reducing the development and clinical risks. So that takes me to the end of our presentation. Hopefully leaves plenty of time for people to ask any questions they might have. I'll go ahead and stop sharing here.
There we go. Perfect. Thank you. Folks, if you've got specific questions, please use the chat function. If you go to the bottom of your screen, you'll see a little icon that says chat. I know, well, I'm going to assume a couple questions, and then I'm going to allow people to sort of take their time and ask them. But Jennifer, the company's announced the intentions of getting uplisted on NASDAQ. Yep. Is there anything you can tell us in terms of potential timelines and progress on that front right now?
Sure, absolutely. So yes, in terms of timelines, we're hoping to accomplish that in the next 8 to 10 weeks. One of the reasons why IPA is able to do that is we do qualify for MJDS uplifting. And then, just already being on the OTCQB already is able to check a number of boxes for us that would normally be oftentimes new requirements that other companies haven't already established, like DTC eligibility and those sorts of things. And so moving forward with that, there's some things that we can't disclose because we haven't addressed, but we do have our team in place. We do have our legal counsel in place, as well as other members. I think it's a team of about 25 people helping us move, safely but swiftly toward that direction. And so right now we're quite on schedule for that, and I think we'll see some pretty substantive steps toward that over the course of the next even 3 to 6 weeks, with respect to some of the things that we need to put out there and make publicly available on that roadmap.
Fantastic, thank you.
Mm-hmm.
So we've got a question here, and it just says the warrant situation. So I know it's obviously public information that we had roughly 9 million warrants that expired here I think it was yesterday. I think at this point there will be a press release, so we would probably leave it at that. There'll be a press release announcing some of the updates on the warrants. There are some warrants, a small number of warrants that'll expire in the middle of October. So I'm sure we'll get updated there as well when the time is right. Jennifer, anything you can share on the diagnostic kits?
I wish there was more I could share on the diagnostic kits. I can share some things, that were released by the University of Victoria, because they have made, progress with the diagnostic kits in terms of being able to validate antibody binding to antigens in order to get a color change that can be read and verified compared to controls, from my understanding, using right now commercial apps that are available on the telephone. I'm not sure whether the plan will be to go forward with a commercial app or a custom app. And so, from that, a lot of the proof of concept with respect to how the test will run has actually been demonstrated. But what we haven't really released yet specifically is the information on the antibodies from Immunoprecise that they will be using. Because right now the antibody in the initial tests that Dr. Brolo has spoken about are what we call surrogate antibody that will do the same thing, but isn't one that will necessarily have the same specificity and ability, the same different types of characteristics we're looking for as we pull the lead candidate out. But as soon as those do become available, we'll certainly let you know. We've got a draft press release ready, and we're just waiting for that data back from Dr. Brolo's lab.
Fantastic. Thank you. Another question here is in regard to government funding. Now, we've received some government funding, and that's been press released. And maybe the way we can't get really into specifics, but safe to say that there's significant other funds that are available out there, both in Canada, the US, and probably internationally. Anything you feel comfortable saying in terms of some of the things that you're seeing out there?
Yeah, absolutely. So, and I think we did release just a little bit of a blurb on one of our previous press releases about this. We are actively engaged in conversations on both sides of the border at the federal level. So you're right, there are a lot of opportunities out there on both sides. They work a little bit differently, but in particular, I wish I could say a little bit more about this, and hopefully we will be able to soon. But but in particular, yes, some of those conversations definitely revolve around manufacturing of the product and clinical trials.
Mm-hmm. We've got another interesting question here. So, what's the opportunity for Immunoprecise on the CRO side beyond preclinical work?
Mm-hmm. Yeah, so on the CRO side, there's just substantial amount of opportunity, actually. That's not something that, with Talem's pipeline, we intend to, let the CRO just slip now or have that just turn into a foundation or a technology base for Talem. We do intend for that CRO side to continue to grow rapidly. both through organic and also continued accretive acquisitions. What we really needed to do as a company was to build an exceptionally firm foundation that could absorb new strategic acquisitions with an integration plan, which wasn't in place before, and without having that in place, it's very difficult to do those integrations seamlessly and to do those integrations in a timely manner that allow you to recognize those cost synergies that are so important. And so we are really looking at continued R&D investments and also some strategic acquisitions. We do continue to R&D out some of our strongest platforms, like our B-cell platform and our bispecific platforms. We always want to stay ahead of the competition. We're always keeping our ear to the ground, finding out what's new, what's next. Is anybody catching up? What's going on? And just pushing the boundaries on that. And also along the lines of automation. And just continuing to automate. We have some things that are still semi-automated. We have new technologies that have emerged that can help us to further automate those. And then also with respect to that, one of the things that we've brought forward, and we continue to say this is, it's unparalleled in our space. We've brought forward everything from concept to discovery with multiple options, so that we can customize these programs for clients, and then also help them figure out, what are their lead candidates? What should they really invest their time in to move forward that has real potential? And then, manufacture those candidates and remove liabilities to make sure they'll manufacture well in the clinical space, and humanize them if they need to be, so they will be less toxic in a human, and even engineer them. We can look at all different aspects of ways we can improve them. But so we bring this forward, and it really has revolutionized things. We've heard from pharma partners, that they've been able to reduce sometimes 10, vendors into, IPA, sometimes up to 27. That's the most we've heard so far. And we've had people just say, this has completely changed the way we do things. But there are still areas where we have to drop clients off. They need to go, we need to tech transfer manufacturing capabilities, protocols that we've designed on their products to a third-party manufacturer to do clinical manufacturing. So that's the, the product that goes into the human. And that the reason why we don't do that part of the manufacturing is that it has to be done under a completely different regulatory environment where everything is monitored. And so, with respect to that, it also makes complete logical sense that we would move into that area, that we would do the preclinical animal manufacturing and clinical manufacturing, so that clients literally never have to leave our door from the time they have an idea in their head to the time that their product is shipped to the clinic to go into humans. And that's a huge benefit also, not just for us and our products, but for those pharma clients, because if we've been working with that product, we developed it, we've designed all of the protocols on how to manufacture it, it's much safer for us to continue with our personnel, our equipment, and our reagents, and the protocol we designed, than to tech transfer it out to another group. So right now, we see a huge advantage in doing that in the CRO, because we can continue to serve those clients, and it's one of the few spaces in the CRO market that actually has a profit margin similar, to us. That's up there. The average in our market right now is probably somewhere around 10 to 12%. We tend to sit more around 60%. So moving into that market also makes fabulous sense, because those are expensive programs and good profit margins, but it also would be, again, just a huge added bonus for our clients, and then something that Talon could tap into and use that manufacturing platform also to, swiftly provide the products that we're producing, both during, COVID, and then also for other Talon products coming through the pipeline.
Some of those sort of GMP, the manufacturing side, some of those contracts are significant. They're not, they can be pretty large dollar numbers as well. So it'd be great for both the client to have that continuity, and for, obviously a provider like us. Okay, another question. I think I understand this one, but for greater certainty, IPA has the capability of multiple cocktail therapies. Now, I'm assuming he means obviously for COVID, but I mean, how easy is that for anybody else to sort of put together multiple cocktail therapies?
Yeah, yeah, that's a really good question. And I, kind of alluded to it as I was going through the deck. But one of the challenges with doing that, with producing a good cocktail therapy, is that you have to, you obviously have to start with a really large program, and a program that's scalable, and a program that's going to give you diverse candidates. And so without getting too technical, our programs allow us to do that because we can pull these human samples plus all different sorts of organisms into these programs and then run them across multiple discovery platforms, again, across the globe. So we can go full speed in parallel at the same time and then monitor it all from a single location. But the key is that, at the end of that, you have to come out with enough potential candidates that you have great diversity of how they manufacture and how soluble they are. Some cannot be actually mixed together. And also understanding, where they bind on the virus, because when you build a cocktail, you specifically want antibodies that bind to different regions of the virus or different regions of a virus protein. And so, with all the different criteria you need, you have to come out of a program with that sort of diversity and ability to also, ideally, put those together in different formats. We have traditional antibodies. We have what's called bispecific antibodies that can bind to 2 different regions on a virus or a protein. There are what's called VHH antibodies, where it's a single chain that can find little nooks and crannies in different places on a protein or virus to bind to. All of that diversity enables you to look at different ways to put those cocktails together. And so, one of the challenges, if you don't have this type of broad program, if you don't have diverse sets of antibodies coming in, so that you come out of it with enough antibodies to start looking at all of these options for cocktails, then it becomes very challenging for people to accomplish that. Because what you'll find is, one of the things that we did is we, in the process of collecting all that information, we set antibodies into what's called bins. And those bins basically represent, which of those antibodies can be put together and enhance the total effect of the cocktail. So we call that the synergistic effect. And so without a vast number of antibodies to be able to test that with, what you'll find is, okay, if you only ended up with 15 lead candidates, finding 3 or 4 that work well together is going to be much more challenging, because in some cases they won't have synergistic effects. In other cases, they'll block each other's activity. In other cases, they would be just challenging to formulate together. And so, with respect to our capability of doing this, part of what allows us to do that is we have multiple, different discovery platforms. And along the way, we can start looking at all those things. How will they manufacture? How will they work well together? Can we limit liabilities? And then the different formats of antibodies. And then again, just going through this rigorous screening of understanding all of that, so we know exactly, which can be put together. On a traditional, platform for discovery, that would be exceptionally challenging to come out the end, even to build a cocktail with 3 or more antibodies, nonetheless multiple cocktails.
Fantastic. Okay, next question. What do Canadian U.S. government agencies need to see before they'd be willing to grant or fund your development or manufacturing? I probably know the answer, but why don't we hear from you?
Yeah, actually, so, with respect to government agencies, obviously part of it is depending on the ask, but, if we're looking specifically to grant funding for manufacturing, the development right now actually is mostly covered. So we've already got that covered through some US funding. But for manufacturing and also for clinical trials, the main thing that they're looking at is the quality of the science around the development work that was done, and the quality of the science of what came out. And I don't think, I can't guarantee it, but I don't think that was the focus 3 or 4 months ago. And I believe what happened is there was a little bit of, flashback and some reflection on some of the initial funding that was done. and a number of different programs that opened up, that really specifically wanted to make sure that they were getting behind the most promising programs. So in our experience, they're not looking for phase 1 clinical trial data. There are a couple pharma groups that would prefer to see phase 1 data before coming in at a phase 2A or a phase 2B, but for the most part, federal funding seems to be looking primarily at, how did you design this? Why did you design it that way? Do you really know what you're doing? Take me through the steps of what you've done. Show me, what your outcome is, your output is, and why it is superior to some of the things that are independent now.
Perfect. Okay, now today you announced a deal with LifeVax, or it's just sort of the start of preclinical trials with LifeVax. What can you tell us about that? And recently you also mentioned a collaboration with Zymeworks. Maybe give a sense of why we work with a company like LifeVax and Zymeworks, and what you expect to see out of this.
Sure. Yep. So LifeVax, the reason that we partner with LifeVax is they have what's called an adjuvant, a proprietary adjuvant. And an adjuvant is something that works to, in a very specific way, stimulate the immune system of an animal or a human that aims at generating an immune system that will enhance the efficacy of the vaccine. So LifeVax had a really interesting vaccine that in many ways seems to be a good fit for addressing vaccines for SARS-CoV-2. So the purpose of partnering with them for the preclinical trial was to assess the efficacy of our vaccine candidates with their adjuvants, and also to see, is there an enhanced effect of protection? When we challenge those organisms with the virus, how well protected are they? Is it enhanced by the adjuvant? Were we better without the adjuvant? But also, it kind of our take all along with the therapeutics, we have a similar thought process here too, is that we're also going to be able to collect a lot of information and understanding how these, the specific variants that we produced for this trial impact the immune system of these animals and how they respond, because that's going to be incredibly informative, and also how they respond in the combination with that adjuvant. So, it may not be the only adjuvant that we trial. In this case, it's very interesting for us to look at some of the differences in the responses we'll see in these initial vaccines we're testing. This is not our final Polytope approach. That this is a different vaccine that we put in based on some information we've gained from the therapeutic antibody work we've been doing, that has given us insight into the particular parts of the spike protein that have been leading to quality functional therapeutics. Now, when we go for the Polytope portion, which will be a separate vaccine that we're engineering, and I mentioned in the press release, we're hoping to have that started also by the end of this calendar year. The difference between those two is, in that approach, we're taking the lead candidates from one of our therapeutic polytopes, and we're currently right now conducting what's called a cryo-electron microscopy study, or cryo-EM study, with the NIH. And what that does is it looks at those therapeutic antibodies binding to the protein, and then it actually gets down to this actually gets really fun, you guys, but it gets down to the atomic level of where that antibody binds on the protein. So the exact amino acids involved, and to some extent even the atoms involved in how the antibody binds to the antigen itself, to the virus itself. And why that's really important is in part it's important because it really enhances your position in a patent, makes your patent stronger. It's fantastic solidifying backup information for publication, but it also informs your vaccine in a very specific way, because you know exactly what the immune system needed to see to make that protective antibody. And so extracting that and knowing exactly what you put into the formulation from the Polytope approach means you can put in the minimum required amount of material. as long as it's in its correct, we call it a conformational form, right? As long as it looks right to the immune system, you can put in a minimal amount of that protein to elicit the same immune response that elicited, the therapeutics that are showing success from the Polytope state. And why that's important is, you don't want to put anything into the human body that you don't have to, not in a vaccine. Everything else that goes in there is superfluous and then poses additional risk when it comes to a safety profile. So we're really excited about that vaccine as well. Not necessarily less excited about this one, but that's the difference between those two. And then the collaboration with Zymeworks. So there were a number of reasons why we wanted to work with Zymeworks. Actually, we announced also that we will be working not only with Zymeworks but also with the NRC. We've been working pretty closely with the NRC in a number of other aspects. They actually provided what's called the trimeric protein that we're doing the cryo-electron microscopy work on. And they developed and designed the protocols, for Zymeworks for the bispecific work. Our candidates coming into the selection for these Polytope antibodies, again, it's a unique and very diverse set of different types of antibodies. And many of those antibodies lend themselves very well to bispecific platforms. And so on a bispecific platform, like the one that Zymeworks brings forward, which is very flexible, interesting to use platform, it enables us to do a couple of different things. It enables us to look at all the antibodies that we're screening in multiple different combinations, because remember, a bispecific is an antibody that binds to 2 different places instead of 1. So we can look at, does that bring an enhanced effect? And we can look at all the combinations, all the antibodies mixed together in a dual combination. Does that enhance? And more importantly, does it actually reduce the risk to the patient? Because again, you're able to control now, again, you're putting in, 2 regions that are going to bind, but you have some control around the remainder portion of the antibody in terms of exactly what you're exposing to the immune system to. And so the hope is that it will bring an enhanced safety profile with those reduced risks and even greater efficacy. But we also are continuing to move forward the traditional antibodies in nature, and don't be surprised if you see, other different types of formats coming forward from IPA, because we have so many different antibodies to bring forward. And our hope is that, we will continue to lead the path here with this Polytope cocktail approach, really demonstrate why it's so powerful and has that potential for increased safety, but also, continue to move all of these different options for cocktail therapies forward and for different formats forward, because they pair well with a lot of our pharma partners' technologies they have in-house. And there's no, when you formulate a cocktail and you say, okay, these 3 work best together, it doesn't mean they're the 3 best. You have to remember that a 4th one might have been a fantastic antibody, but it doesn't work in that cocktail. So the potential of what we could bring forward and the number of ways that we could partner, bringing those forward, to us is just really exciting right now. But that's why we're forming some of these relationships, both to enhance the technology, to look at reducing, safety concerns and enhancing efficacy, also to bring along some partners that have, a great experience in testing and trialing these different formats.
Jennifer, really sounds like, and I'm going to refer back to you here in a second, but it sounds like we've done a lot of sort of front-loaded work to try to get as much safety and efficacy information before we go into any sort of animal or human trials. Is that sort of the way to describe it?
Absolutely.
Yeah.
Absolutely. And that was our goal from the beginning, going back to, a little mention here also in the slide deck, we saw right away some challenges from the financial push and the political pushes to get repurposed things out there, to get, platforms out there that really didn't have a lot of safety data, in some cases no safety data out of clinical trial formats, and a lot of incentives, financial incentives for a company to push those boundaries. And our challenge with that is that addressing emerging infectious diseases, especially RNA viruses, is not easy. This is not a Hail Mary pass. In a lot of cases, I sincerely think that a number of groups were just hoping to get a platform into the clinic so that they could demonstrate, some safety profiles and have some clinical safety profiling attached to that platform. And there was a lot of financial and political incentive to be first. And the reality is that there's a very slim chance that what gets out there first is going to be what stays there, right? There's also a slim chance that we'll have just one option for a vaccine or a therapeutic. But really, what regulatory officials are going to get behind and what they're going to support is what brings the highest amount of safety with the lowest risk, right? And that's always the goal. That's always what we call our risk safety profile that gets reviewed by regulatory boards. And so, again, I always say, I hope something comes out there that reduces infectivity and can help reduce morbidity and mortality as soon as possible. Absolutely. But I do think that ultimately what is going to bring that for the long, haul and really be the answer is something that has been, designed with this type of rigor and with this type of approach without cutting corners.
Mm-hmm. Awesome. We've got a quick question here in regards to acquisitions. Maybe I'm going to phrase it this way. Obviously, the company's going to have a little bit extra money with these warrants. As far as any kind of acquisition strategy, is there technologies you'd like to see that you add to the business? Is there sort of competitors that you would like to acquire? Or just give me a sense of landscape where you would focus a little bit more of your attention as far as M&A.
Mm-hmm. Yeah, that's a great question. We're always on the watch for, good technologies or disruptive technologies, new things coming up. Sometimes those are in the lab. Sometimes there are other things that could enhance, how we address, especially the discovery part. And so, there are a number of companies we've been watching for some time. Some of them kind of take care of themselves by just making bad decisions and they come off of the radar. But we do have one or two, that we've watched over time and developed good relationships with and have a good understanding of, in particular, understanding what their strengths and weaknesses are and why they have those weaknesses. And so, with respect to technology, yes, ideal situation there, a group that brings forward a technology that is, innovative and has a ton of potential. And maybe this isn't recognizing that potential due to poor management. That's usually the case when you find that, that occurring. So we're not ruling that out. And again, we have a couple of companies we've been watching, but I think it's more likely that additional M&A activity would probably take place back around the preclinical and clinical manufacturing side. And then there is one area that we just haven't announced publicly as a potential M&A activity, but it's just a little bit outside of technology and manufacturing. It's a little bit different, but we really think would just be one more way that we carve ourselves out in the market in a really unique way.
Okay, folks, I'm gonna apologize in advance. And there's a number of questions that we've got here, but I don't think time's going to permit everyone here. So I'm going to quickly sort of zip through here and try to catch them that I think is a little more important. First off, you mentioned NRC, and just so everybody knows, NRC stands for National Research Council, and that's sort of a part of the Canadian research, sort of government research agencies. Okay, so we covered acquisition. Has Talum COVID mission? Okay, well, has Talum rubbed our pharma clients the wrong way? I've had this question asked me a couple times. Maybe discuss how Talem and the CRO business, really, there's a bit of a Chinese wall there.
Yep. Yeah, no, I mean, that's a great question. That is a great question. So, no, although this is not a common model, I have been involved in this model before. So let me just first put some reassurances in that there's a very good understanding of how to make sure that there are not conflicts of interest between Talem and pharma partners. Talem acts as a independent company that is a full arm's length contracting out to our subsidiaries. And so, you know, I won't have time to go through it all because we have very, very strict protocols in place that make sure that we also protect ourselves here as well. But for instance, you know, if we do have, for instance, you know, a target in the lab that we know a client has recently, you know, brought to us, we don't address or touch, you know, that target. But that being said, you know, it's not uncommon when a hot topic comes out and gets published in Nature or Science that, you know, our phone starts ringing and 5 or 6 clients are going after the same target. So we have many of the same protocols built in place to protect our clients from their competition as well, not just Talem. And so, you know, without getting into it, because it's not maybe the most important side, but just briefly going over, we never share ideas ever. If a pharma client has a unique idea that is not publicly known in the space that they think is going to enhance their program, that never gets shared. It never gets put in someone else's program, and we would never use that idea. The same goes for Talem. You know, if Talem has a great idea and a way to advance the product in a way that, you know, is not publicly known, Talem doesn't have to share that idea with the CROs either. They are restricted to respecting that confidentiality as well. And so, you know, every company is treated, you know, very strictly like a separate company, including Talem. And that also goes even for very basic things. You know, we make a reagent, you know, for a program that we use in the very beginning of the program, and then we're done with it. And we don't stick it in the freezer and pull it out for another client. We start all over, make a whole new, you know, product for that client. So we don't overlap on reagents. We don't have their work in the same lab on the same day. So we can also avoid any sort of confusion along those lines. We have very strict protocols for documenting as well. So in terms of how we rubbed them the wrong way, actually, I think it's been exactly the opposite. So, you know, most of our clients, it's very much based on relationship and trust, right? They know us, they trust us. They build these relationships with their project managers over time, but also, you know, we can't also overlook the fact that a lot of our executive management team has been in this field for 10, 15, 20, 30 years even. And so they've built relationships with these decision makers at pharma who trust them and know that, you know, that they can trust that individual and have this relationship built. And so, you know, when it comes to, you know, working with those pharma partners, actually many of them are quite interested in what we're building in Talem and wanting to be kept updated and wondering if they can sign CDAs to get a look at the targets and understand better, because it's much better for them if they come up with an idea and we've already initiated something in Talem and we're 6 or 8 months down the road. And so what we're finding is that actually most of the interest in Talem assets is coming out of existing partners, because, and honestly, I think because they understand our technology, they understand the quality we put behind things, and so they know that, you know, they can leapfrog ahead with probably, you know, a very similar design of what we would have done for them.
Excellent. So we obviously have someone who's got some basic science background here, because this question has words that I cannot properly probably read here. Okay, how can you be sure that your vaccine generates an immune response against all varieties of COVID-19 antigen types?
Mm-hmm. Yes, so great question. So the vaccine itself is based on the actual epitopes. So again, we have multiple vaccines, but the strategy with the Polytope vaccine is that we're building it on multiple epitopes to represent the epitopes bound to by the antibodies that have been selected for the Polytope, right? And then behind that, so it won't cover everything with one vaccine, right? The key is that because it's addressing multiple epitopes, so if you've got the 3 antibodies in the Polytope vaccine, for instance, or the Polytope therapy. The Polytope vaccine covers 3 separate epitopes. So it is the same philosophy here. If you look at when a virus mutates, if you have a single amino acid change, so 1 mutation in a place where that antibody binds to, that antibody will no longer be able to bind there, possibly more than likely, and now that antibody is no longer efficacious. And so it's really risky to put a single antibody in. You put 2 antibodies in, and now you have a single mutation that occurs. 1 antibody is no longer useful, but you lose the synergistic effect between the 2. And now your whole therapeutic is hinging on 1 more mutation not happening. That's why when you go to 3, you have the added protection. It's the same thing with the vaccine. The vaccine is putting in, a minimum of 3 epitopes. Now, if you're vaccinating against that and the virus mutates on one of those epitopes, your vaccine is no longer protecting you in terms of having your body generate antibodies to that particular epitope. But the other 2 in the vaccine are still generating immune responses within the human that are going to be protective. So it would require 2 additional mutations in those regions of the epitope in order to render a vaccine ineffective. So it's much more powerful than putting in a single epitope unit. And going back to the idea that, yes, could you put the whole darn, a whole protein in there, right? And maybe, then you're just covering lots of epitopes. Yes, you could, absolutely, but you're putting in all of these other regions on the protein or on the virus that are not going to be protective and may even elicit undesirable immune responses, whether it's cytokine storm or whether it's immune responses that are just very powerful and not protective and outshine or outshadow the performance of antibodies to epitopes that would be more protective. So I think that's a key part. But to address, when I say that this can be protective in the longer run against variants and strains of the virus. What we're referencing is that with the Polytope version of the vaccine and the therapy, say if we focus on that vaccine and you have a mutation in 1 epitope, so the other 2 are still protective. So you're fine for the season that you vaccinated everybody for, right? Or maybe it doesn't become seasonal. You're fine for a while for that vaccine that you used to immunize and protect everyone. But now remember, we have all these other antibodies that we collect epitope data on, that we have all this information on. So all we have to do is say, oh, well, it was amino acid number 126 that switched from this amino acid to this amino acid. This 1 component of the vaccine is no longer going to be protective. We still have 2, that's fine. Now we go to our database and we pull out a 3rd one based on the epitope that we know provided a protective antibody. And that one gets plugged in to the new vaccine. And that's how we plug and play. We have the safety that the 3 epitopes in the vaccine are going to provide us, long-term protection, and give us the time to be able to pull one of our additional epitopes in to reformulate it and continue to provide protection. And so that's why it's, the broad comprehensive approach for both the therapeutic and the vaccine are so important, not just for creating something that's initially safer and more efficacious, but something that's going to enable us to put a vaccine and a therapeutic out that will have sustained efficacy. And if something changes, we should have plenty of antibodies in our repertoire that we've already mapped, we already understand, we already know which ones can fit together. Remember I said we created those bins, so we can look and say anything out of this bin should work. We pull it out and reformulate. So it's a huge advantage going forward. It's not just about the initial vaccine and therapeutic. It's also about the collective data that we now have.
That's amazing and exciting, Jennifer. And so this is going to lead into this other questions, but obviously the experience we're gaining on COVID, does it give us a head start in the event that there's another pandemic?
Absolutely. Mm-hmm. Absolutely. And that's the entire intent of building that database. And, well, I should qualify that. So a new pandemic with a different emerging infectious disease, mm-hmm. This would be a little bit different, but yes, it absolutely does that too. So what we've used here, so obviously I think I've outlined here how it helps us with a new pandemic with a new strain, right, of coronavirus. But in a new emerging infectious disease situation, absolutely. So one of the things that we've done is we've created templates as we've gone through. So we have all the ongoing modified templates of exactly how we address coronavirus. And then we also have taken that and, you know, whittled it back for when we do need a rapid response. In some situations, we will need to be more rapid. And, you know, for the person who asked the questions about the grants, that's a great example. I wouldn't, for instance, go knocking on the door of the DoD or the armed services and say, hey, by the way, you know, we're going to do emerging infectious diseases for you, but on the, you know, on this sort of timeline, they would really need something rapid. But you still have to put the power and strength of what we're doing behind that. And so our ability to go through and say, yes, this works great. You know, here's our protocol that we launch as soon as something like that happens. And here's the areas where we can, you know, shrink this so without cutting the efficacy or the safety profile. But in those cases, then, you know, we would probably need to look at a different route for manufacturing, probably sponsorship right in the hands of a government group like that. But it really has also enabled us to take a look at that and address, okay, you know, going forward, here's our template. Now, exactly, you know, how do we use this template? And this is something we're in active conversation about right now. As soon as another emerging infectious disease comes up, what have we learned from this? What could we do slightly differently? So for instance, neutralization assays to determine what antibodies actually protect against the human cellular invasion. That's not something we were set up to do, right? So can we bring that in-house, and can we do that faster than the group that we use? We also now have all these great partnerships in place, right? So groups that, you know, we didn't bring in for asset ownership, but that, you know, partnered up with us for regulatory questions, for, you know, what's called CMC questions, helping us understand how will we formulate the final vaccines. You know, those are groups that we didn't have on board and a good, strong working relationship with. So there's probably too many ways to list it, but absolutely, we feel very prepared to use these platforms, which we really think are ideally situated for emerging infectious diseases going forward in those types of scenarios.
Mm-hmm. Great. Okay, two more questions. I'm going to ask, so a lot of these assets that Talem is building, the goal obviously is to push these as far as we can to the clinics and effectively commercialization, but there obviously also is the opportunity to, you know, a biovac deal, or should a big pharma or some entity come along, that's something the company's looking at doing, as well as to entertain some form of partnership or co-ownership. What does something like that look like in this industry?
Yeah, so that's a great question, because it always can look a little bit differently, and you're exactly right. There are pharma partners who look at our different assets from different perspectives and see, oh my goodness, this fits with their technology over here. Different type of delivery, different type of formulation, or this technology over here that really focuses on something key to SARS. And so, you know, yes, we are looking at moving many of these things forward on our own, but yes, we're also involved in those conversations, and with, you know, with large pharma sponsors. And there's huge benefits to that, because we don't currently have a commercialization team. And those teams are really important for understanding those bits and details in you know, everything after you know, getting through the clinic. And so, you know, what do they look like? I've seen a little bit of everything. I've seen people come in and you know, license those types of products with a specific endpoint only. You know, do a licensing deal for an end product that says we're going to pay you X amount of money all up front, you know, just to use this, for instance, in phase 1. And then they would have to come back and negotiate. There are other groups that come in and say, all right, we're going to pay a certain amount of money up front, but we're going to backload it on milestones. So if you're successful in phase 1, if we're successful in phase 1, we'll pay you this amount. If we're successful in phase 2, we'll pay you this amount, etc. So kind of a milestone based. There are other groups that would rather pay just a larger amount up front and then do something like a royalty after commercialization on the backside. So we've really seen a little bit of everything in this space, not really necessarily pertaining to coronavirus because it's new, but those would be very typical examples for therapeutic deals being struck in other disease indications.
Fantastic. Okay, so last question, we'll leave with this one, but in the next 10 years, based on what you see happening in this industry, where would you expect IPA to be 10 years out? What would it look like? What would the company potentially look like? 10 years is a long time, obviously.
It is.
But give us your long-term goal in terms of what you're going to do with this company.
Okay, all right. So really focus on the forward-looking statements because I'm just going to give you my vision, right? I mean,
awesome.
Yeah, okay, so what I really think this could be, I think this, if I was going all out and said, you know what, we're gonna make everything happen, this is really where we could be in 10 years. We could, for instance, have our own transgenic animal lines that are fully human. So we're not only in the CRO space, but we're, you know, creating licensing and royalty deals with pharma companies to use those, but also just, you know, really, you know, again, you know, changing the way we work with pharma partners who currently still need to negotiate that. And we've made a little bit of a move like that already, obviously, with Ligand Pharmaceuticals in Talem. And that CRO part of our business, I honestly think the sky's the limit with that. And again, you know, qualifying that statement, but I mean, there's so much potential there right now. When we say, you know, this is the market we're in, the therapeutic market, what the heck is the therapeutic antibody CRO market? Right now, it's like thousands of little companies, right? So you have all these companies that do antibody discovery, and you have companies that help with your lead development and characterization, a lot of them just in characterization. Some that specialize in just doing the functional testing and understanding what exactly the antibody is doing. You have others that engineer them for you, others that do your humanization. You have a ton of companies that do manufacturing. So really, and that's part of what I think personally leads to the lack of innovation. They're small private companies where in order to innovate, that money's got to come out of the owner's pocket. So there's always an internal dilemma on, you know, how much personal money to reinvest and how innovative to be. So in a you know, ideal situation here, we just get, I mean, if we're doing the best that can be done in characterization, in discovery, in engineering, if we're bringing forward the best technologies, there is no reason for all of these companies to continue to exist. If we're able in our discovery platform to go for regular antibodies, bispecifics and trispecifics and VHHs, and we can go after antigens that have sugars on them in just the sugar group, or go after a phosphate group, right? A single chemical modification. There's no reason for them to have all of these different groups that they're using. And then again, you know, so we, you know, ideally, we absorb all of those markets into one and the downstream GMP manufacturing. So when a client comes to us, you know, we move our own programs through, instead of saying, here's your target, here's your plan, here's how we're gonna lay out getting you from where you are right now to, you know, a product you're ready to go, you know, and apply it for an IND application with. You know, we hand over everything, the regulatory proposal and how to move forward, the manufacturing proposal, we start laying out right along the way, side by side. You know, how are we going to move this into, you know, engineering runs for clinical manufacturing? We're helping them build their IND portfolios for the government regulatory authorities. And we're moving them, you know, into that GMP and through, and then that same pipeline is what's being used for Talem. And I really think, you know, Talem is something that we will continue to invest in heavily. My hope for Talem is that assets fly out of there faster at some point. You know, they need to mature a little bit. COVID's probably gonna, I hope, COVID will go a little bit faster than the other assets. Some of the other assets just need to mature a little bit. And some of them slowed because of our focus on COVID. But my hope is that those start getting out licensed and bought so quickly that we just have trouble coming up with enough to keep that pipeline full. You know, that's definitely, you know, that's definitely a goal. And I think when you know, when we look at envisioning where could we be, we can look at companies like Charles River, right? I always look at Charles River when I picture that, because, you know, 10 years ago, 15 years ago, small molecules was everything for treating diseases, right? I mean, antibodies were coming along, but the majority of treatments you received for all of these same types of diseases were small molecules. And now antibodies have just completely overtaken that. And they are the #1 type of treatment you receive for the majority of these chronic diseases, such as, you know, cancer and inflammation. And they make up the majority of the revenue in that commercial space. And so, you know, when you look at Charles River, they, you know, go back however many years, 10, 15, 20 years, they saw this very similar vision for small molecules, and they put it together and they accomplished it. And then look at where they are today. It was very much end to end. And it was very much helping the client through that entire process. I'm not so sure as much along their own products as it was focusing on animals. And I think that's a good thing to envision. It's a helpful way for you to envision what my vision is, you know, for Immunoprecise and where we could be. We'd be the end answer, the goal for all pharma.
Fantastic. On that note, Jennifer, I think it's a perfect spot to end. Certainly on behalf of everyone, I know I want to thank you very much for the time that you spent with us. I know you're very busy, lots on the go, and hopefully you get a chance to relax a little bit this weekend. But yes, lots to look forward to. Obviously, I'm pretty excited with how things are happening. And I want to thank everyone today for listening in. I want to thank Jennifer one last time, and Trevor. Unless there's anything else you want to say, Jennifer, we'll hand it over to Trevor. And we'll
Sounds good. Just to say sorry we couldn't get through all the questions. They're great questions, and really appreciate everyone, you know, coming to hear more about IPA today. It's a pleasure to be able to share.
What's the website for IPA?
Immunoprecise.com.
Perfect. Okay, lastly, well, again, just thanks everyone, and everyone stay safe, and we'll talk to you guys all soon. Trevor, over to you.
Thanks again, Paul. Thanks, Jennifer, for joining us today. And we're looking forward to the bright future that IPA holds, and looking forward to that NASDAQ listing for you guys. And thanks again for your time.
Absolutely. Thank you.